Solving the Pandemic, Episode 1: How to Leverage Existing Clinical Trials to Solve the Pandemic. Hi, my name's Dr. Elizabeth Bent, and I'm a molecular microbial ecologist, which means I study DNA or RNA from samples of material to see what microbes were living in them. I thought I'd share with you some thoughts and information about what researchers are trying to do in the world to find a solution to the current pandemic, treatments or vaccines that will allow everyone in the world to go back to life as normal. I hope this series of podcasts brings you some hope as well as some new information. The main reason I wanted to start this podcast was that I had an idea. On March 28, 2020, I was thinking about clinical trials in the context of some research I've done with the gut bacterium, and it occurred to me that the strategy I came up with would be useful not only for clinical trials with that bacterium, which are in progress at the moment, but with anything that is currently being assessed in clinical trials, anything at all, any medical treatment at all. The idea is really simple, yet it's really powerful, and I'm going to focus on it in this first podcast episode. In future episodes, I'll talk about how clinical trials work and the kinds of research going into solving the pandemic that are ongoing. But for this episode, I want you to listen to my idea. If this podcast is listened to by a clinical research scientist, I want that scientist to hear this idea right at the beginning of this series. It may be the single most important or useful idea I've ever had. The idea is this: at the very end of clinical trials for a drug or any other medical treatment, a great many patients are usually enrolled, and this can number in the thousands. This is because at this stage, researchers have already verified the safety and the efficacy of the treatment, and now they're looking for unusual side effects which may only affect a small proportion of the population. To find the effect, you have to look at a lot of people, so they enroll a lot of people at this stage in the trials. So it occurred to me that COVID-19 infections are widespread. They are really, really widespread. It's unlikely that clinical trials currently underway at this final stage have all their patients avoiding infection. So why don't we use this? What if you took an existing clinical trial in progress, late-stage trial with maybe 1,000 patients or more, and assessed each patient for COVID-19 infection and symptoms? Then you could identify a subset of patients in each treatment which are infected. You can use standard statistics to contrast the effect of the treatments on COVID-19 infections, and your results would not necessarily be skewed due to a tiny number of patients because the trial had so many to start with. You could determine from this kind of data if the treatment suppressed symptoms, or alternately, does it put people at greater risk of developing symptoms if they have the medical treatment in the trial? Because you have not only the treatment, but you've got controls. So it's not an ideal situation since the data set, the experiment, wasn't carefully designed with the maximum number of patients that are all exposed simultaneously, but we need solutions to the pandemic sooner than later, and I think we all can agree on that. If this kind of analysis can provide some hope or point to potential treatments that can be used, or alternately, additional risk factors that we don't yet know about, it might well save us all a lot of time. And unfortunately, people are already jumping to false conclusions based on scanty data, so a larger trial that's currently in progress with more patients should do better. For example, the French study done with hydroxychloroquine and azithromycin that's been mentioned a lot in the news and even by US President Trump pointed to a treatment that people thought might be useful even though the number of patients was very small, and because of this, the study had to be considered very preliminary. The citation for the French study is Gautret et al., and it's published in the International Journal of Antimicrobial Agents, article number 105949, and it was published on March 20th, 2020. It's worth noting that these initially promising results have been refuted in a second paper published only 10 days later, on the 30th. Molina et al. In Medicine et Maladies Infectieuses, the title, No Evidence of Rapid Antiviral Clearance or Clinical Benefit with a Combination of Hydroxychloroquine and Azithromycin in Patients with Severe COVID-19 Infection. So this situation is why it's so important not to jump to rapid conclusions when testing a drug or a combination of drugs based on a small number of patients. Because of the publicity surrounding the first paper, people have been self-dosing and harming themselves, and other researchers are more thoroughly investigating this treatment, which might not actually have any real benefit. On April 6, Rutgers University in New Jersey announced it was starting a clinical trial of azithromycin plus hydroxychloroquine with 160 patients enrolled. So let's hope that those patients do well and not suffer. Getting back to leveraging existing clinical trials, Perhaps an even greater number of patients can be assessed through larger-scale late-stage trials of other treatments that are already in progress, in which a substantial proportion of patients are verified to be infected with COVID-19, whether they're symptomatic or not. There's no ethical issue because the patients were accidentally infected, and at this stage in the clinical trial process, the treatment has already been vetted for general safety. So something that's being tested that's out there might help provide clues, if not an outright solution, as to how to treat the pandemic. And all that's needed is some extra work, one more step. Even if the probiotic bacterium I studied, which is currently being tested in late-stage clinical trials as far as I know, could be helpful, and I can explain why I think so in a later episode. For now, I just need people who are running trials to look at them, and even if all you uncover is an additional way to identify people at greater risk of developing severe symptoms, you will have done something extremely useful. To deal with the fact that patients were not simultaneously infected in a controlled manner, it might be possible to use epidemiological data to model when patients in different areas were likely to have been exposed. If the only thing that gets shared from this series of podcasts is this idea about leveraging existing clinical trials, then I will have considered this series a success. I have tried really hard to get this idea quickly to the research community in a variety of ways, and it's actually kind of difficult. I can't publish a proposal paper or a hypothesis paper to a public archive, like the medical archive, medrxiv.com, although I did try, and my attempts to reach scientific organizations, health ministries, newspapers, and people within my small circle of acquaintances who might have connections to clinical research scientists have largely failed. So I'm relying on you, dear listeners, to help share this idea and get it to someone who can spread it to the right people in the research community. I've detailed the saga of my thought journey and the difficulties involved in advertising this clinical trials idea in my personal science project blog, www.gut-bacteria.com. I started this blog in 2014 for a crowd-funded effort called Project Cancer Fighting Gut Bacteria. This project has merit, but it's been stalled due to some difficulties. I'm a microbial ecologist in Canada with no laboratory of my own. I'm trying to fund work that would need to be done by more medically oriented colleagues in the USA, and I'm trying to use byproducts from a now patented bacterium from the University of California. So I've largely given up on trying to find funds to test the hypothesis I've developed out of the preliminary work I've did, and I've recently decided to just publish the hypothesis, so that's coming next. I've also done a small set of data analyses based on my hypothesis using my 2014 data and some human pro-inflammatory cytokine genes that are important in the cytokine storms induced by COVID-19, the runaway inflammation symptom which kills people. I wanted to publish my thoughts that are inspired by all this, as well as the leveraging existing clinical trials idea really quickly to an archive so that people could see the clinical trials idea at least and work on that. But as I said before, the archive I tried does not accept proposals, and this clearly would only be a proposal, not finished research, since I'm not conducting any clinical trials. So while I rethink how to package my thoughts into a more acceptable format for rapid publication and sharing in the science community, I am trying this unorthodox method to reach the right people with this podcast episode. So if I could talk for just a brief moment about the sorts of work that you would have to do if you were a clinical trial scientist and you had a lot of patients enrolled in your trial and you wanted to try this idea, you might not even need to call in all your patients for an assessment and new samples if you have archive samples that can be used, blood or throat swabs or saliva or even fecal samples. If you took a subset of those and you extracted RNA, if the extraction of the RNA is a problem for your laboratory because you don't have the right facilities or whatever, I have a few ideas as to how to make it safer and more rapid and cheaper in my my science blog, www.gutbacteria.com. So take a look at the March 28th, 2020 post towards the end there. I have some ideas, but anyway, so the idea is that you take your samples, your archive samples, you extract RNA from the virus, and you obtain it in a form that's not going to be infectious so that the technician can handle these samples safely. And then you do quantitative PCR, which is a method that can quantify the number of initial RNA sequences in your samples using CDC primers that are already been designed for this purpose that are available on the CDC's website. So you can do quantitative PCR, get the viral load in your samples, and then you can figure out who's been exposed to more virus and who hasn't, and who's, and as for the symptomology, a simple questionnaire should be able to figure that out. So it's not necessarily going to be a huge major pain in the butt to try and get a lot of people assayed for COVID-19 infection. So, my dear listeners, if the only thing you do to help with the pandemic is forward this episode or information about it to people who might know people who conduct clinical trials, you could be doing us all a really big favor. In my next episode, I will outline the entire process involved in running a clinical trial, from the pre-trial work that has to be done with a given drug or treatment to the kinds of regulatory requirements that need to be done in order for a trial in human beings to proceed at all, and why clinical trials are done in stages. I already touched on that a bit. It's because it would be unethical to use a potentially dangerous or non-efficacious treatment on a lot of sick people at once. In future episodes, I hope also to describe current work being done to find treatments or vaccines for COVID-19. I'd like to describe how current FDA-approved drugs are being assessed for their potential to treat COVID-19. I'd like to talk about current vaccines that are under development, and I'd also like to talk about how supercomputers are being harnessed to find solutions. I hope you found something useful in this episode, and I look forward to recording the next. In the meantime, stay safe and at home whenever possible. I'd like to thank Matthew Rice for the graphic that accompanies this podcast. This episode was written and recorded by me, Elizabeth Bent. My science blog is www.gut-bacteria.com, and I'll be posting episode scripts such as they are once episodes become available online. Thank you for listening, and if you found things of value here, please remember to share.